Thank you for the introduction. And hi, everyone. My name is Ercag Pince. I'm a postdoc at Roland Institute at Harvard University in Tata Lab. As the title suggests, today I'm going to talk about the role of motility in bacterial coexistence. Bacterial cells have the ability to explore their surroundings by swimming, and they can sense if the life is getting better or worse at the single cell level. They can also form actively swimming colonies and forage collectively. Here you see an image of a single colony propagating from a central inoculum and occupying the niche inside a soft agar assay. One can increase the number of such species and see that they collide to each other while swimming, and then this population can reach to a steady state after depleting the available nutrients inside such a complex medium. As we go further towards more complex systems, we see that the population dynamics actually gets much more intricate. The perfect example is the human microbiome, which harbors a large diversity of bacterial communities that can be maintained over larger length and time scales. So a recent study has shown the switch, or in other words, the transition from a single cell motility behavior to a single colony well. However, despite its ubiquity of motil phenotype found in nature, we still don't know yet how multiple colonies, in other words, multiple motil strains are interacting with each other, and how this leads to a diverse set of bacterial communities. Here we pose the research question: what is the role of bacterial motility in coexistence? To address this question, we focused on a model system based on two natural E. coli isolates, which were extracted from the same habitat, in other words, from the same host. They were living and coexisting in the same gut of the animal. I call them strain and strain B. We transformed these two strains to express YFP and CFP. And the fundamental phenotypic difference between these two strains is that A can grow faster than B, and B can expand in a soft agar assay faster than A. Then we mix these two strains to set our competition assay, and then we inoculated them into either a shaken liquid where the nutrient influx renders the landscape such that the nutrients are always available to both strains. And in the other competition assay, we actually inoculated the cells to a static gel where both strains must be able to swim away in order to reach and occupy a certain niche. How did we quantify the competition outcome? In other words, how did we determine who wins over whom this competition? So we first set the initial population abundances A0 and B0. Then we mixed these two populations, and then we calculated the initial ratio. And once the competition ends, we quantified the final abundances of AB by using our newly developed integrated fluorescence microscopy method, or colony counting. And then we set a metric in order to determine who wins the competition over the course of this this phenomenon. So this parameter is above 1 if A is winning over B, and drops to below 1 if B is winning over B, or B outcompetes A. So it's simply the final ratio, final ratio over the initial ratio. So once we finish this experiment, we check the fold change in ratio for both cases. In the well-mixed habitat and both in the spatial habitat, we see that the system actually follows the competitive hierarchy. So the fast grower dominates over the fast mover in a well-mixed habitat, as I mentioned, where the nutrients are constantly available. But on the other hand, in the spatial habitat, we see that the fast mover, the strain that is able to expand faster, actually proliferates higher than the other one that is lagging behind. And For the well-mixed case, again, if we shift the initial frequencies, we see that the A dominates regardless of initial abundance. This is also kind of expected because as competitive hierarchy dictates, A grows faster than B and depletes the whole, able to deplete the nutrients available much faster than B can. On the other hand, in the spatial habitat, we see an interesting dynamics where the competitive hierarchy is broken. Each strain becomes fitter when rare, and regardless, depending on its initial frequency, both strains can become fitter when they start as a minority. So why is hierarchy inversion, or the fancy name, the ecological fancy name for this phenomenon, is important for coexistence? Suppose we play this game continuously instead of discretizing the initial abundances. So suppose that each final ratio enters to the next round of competition as initial ratio. If we follow such a dynamics and logic, we see that the system actually evolves and converges to a stable fixed point where none of the strains can invade to each other. So this can be ensured by the breaking of competitive hierarchy, and both strains can coexist in such a condition. In the other case where A can dominate regardless of initial abundance, we see that the dynamics doesn't allow B to be fitter even in the majority or minority case. After this, we investigated why the hierarchy is broken at the first place. To do this, we looked at the at first we looked at the dynamics of monocultures. In both cases, A and B can actually occupy the whole petri dish, and they can expand until the nutrient is depleted in the medium. However, when we looked at the structure, special structure, when A and B mixed equally, we see that the A actually occupies the central region, which is closer to the inoculum, and B fences the A around a certain perimeter and impedes with the growth of the fast mover. So as a result, we obtain a dot and donut pattern where the fast grower simply holds the central region and the fast mover actually creates a niche itself around the fast grower. And you can see here the time evolution of the radius of each colony, and we detected the fast mover can cut the nutrient access to the fast grower after roughly 1.5 days and force it to arrest this growth. And after that, we actually asked the question if these two parameters, motility as expansion rate and also the growth rate, how they are influential in the outcome of competition. For this, we engineered a set of strains which are identical except their expansion rate and motility rates. At the first case, where the expansion rate and motility rate are comparable to each other for lab 1 and lab 2, we did not observe any spatial segregation and dot and donut pattern. They lie on top of each other basically. And as we go and ramp up the expansion rate of lab 3, we see that the lab 4 is confined to a smaller region of space, whereas lab 3 actually is able to expand farther. And we recovered the same dot and donut pattern once we increase the growth rate and expansion rate difference between these two strains. And then we see that there is a fundamental trade-off between motility and growth, so that the segregation occurs. We also tried the same experiment with lab 1 and lab 5, where the growth rate and motility, in other words, the expansion rate is much larger. We also explored a Keller-Siegel model to understand the underlying dynamics of this segregation, and the model captured quite well and accurately how the experiment unfolds. You can see that here with, this, with these two parameters changing in the phase space, we recovered the same dot and donut pattern across different lab strains. And we asked the question, if this mechanism is limited to lab strain or is it particular, can we actually generalize this to other natural isolates that also shows a certain growth motility trade-off? The answer is yes. And we looked at the other cohabitant strains from different hosts. And we see that once there's a clear difference between growth rate and expansion rate between strain pairs and between strains inside the pair, sorry, we see that similar patterning occurs. Here you can see how fold change in ratio can actually unfold in a large dynamic range of initial ratio. The solid line shows the simulation outcome, and the markers are showing the experimental results. So we actually tested the larger dynamic range of initial ratios, in other words, initial abundances for strain A and strain B, the natural isolate, and also lab 1 and lab 5, and then we recovered the same coexistence dynamics as mentioned in the previous slides. So just a summary. Just to summarize what I talked to today, we studied the competition between two multinatural isolates, and we see that these two isolates can actually segregate into two distinct niches, one proximal and one distal. And we observed that the competitive hierarchy is broken down in spatial habitats, and each strain becomes fitter when rare. So the selection occurs negatively dependent on the initial frequency. And we see that there's a fundamental trade-off between motility and growth, which can lead to segregation and coexistence. And as I mentioned, we captured the model, in other words, captures the essence of the niche partitioning and coexistence in two-strain model. So as a follow-up, I'm asking the questions: how can two strains with different swimming patterns, like, for example, E. coli, which swims in run and tumble motion, and where Vibrio fischeri can swim in run and reverse motion, can coexist by the same mechanism? So we haven't tested this yet. Can three different strains with different competitive abilities coexist in a special habitat? And also, one particular follow-up question is, how does the shape of growth motility trade-off change when more than two strains are involved? If we increase the number of involved strains, does the motility growth chart will evolve to comprise a Pareto front? That's also something that we are curious of. So I would like to first thank to my collaborators, Sebastian, Katja, Anna-Bart, Sander, and Tom. And I would also like to thank to Emimi for inviting me to this talk. And so I was supposed to give a poster first, but and now here I am. So, and thank you for listening. I'm happy to
thank you. Yeah, thank you very much. There's a couple of questions in the Q&A. So the first one is, when you're looking at the distribution, are you completely sure that there's no any cells from the fast grower at the edge of the plate, or is it completely excluded? Like, how can you be sure that there's?
It's completely. We also tested this by basically single colony counting, but our methods, our integrated fluorescence methods, shows us that there is no leaking or stray cells in the other regions. So they are the segregation is quite robust and quite sharp.
All right. And the other question in Q of A from Bram Van Dish right now is, would strain A eventually win if you have richer media? So can you change the media so that food is still available after B swarms away from the competition? And would that change the dynamics?
That's a great question. Actually, I tested this for the natural isolates, and indeed it is the case. So, and maybe you also stumbled this upon Chen Liu's talk, that the ingredients of the medium actually changes the speed of the chemotactic wave. And that alters the outcome of competition quite drastically. So just to give a specific example, I had a different outcome when I actually implemented this experimental condition in defined ridge with respect to TB medium. So it all boils down to the shape of motility growth trade-off. So how if you map the motility and growth phenotype into two axes, if the shape of the trade-off curve changes, this also changes the competition outcome. Thanks for asking this question.
All right, so we have a couple more questions that I'm going to post to the Rocket Chat channel because we're into the break time, so I don't want to take people's break up too much. But thank you again to all the speakers for the great